Hey there, Science Explorers. Welcome to today's lesson of super quick catalysts. Our science objective today is that Science Explorers will learn that catalase, which is found in plants and animals, breaks down hydrogen peroxide. So we have two essential questions today. First essential question is, what is a catalyst? And the second one is, what is a chemical reaction? Well, if we look at our essential question number one, what is a catalyst? Catalysts are substances that speed up a chemical reaction. And our second question, what is a chemical reaction, is what is a chemical equation? Chemical equations are symbols that represent the change that occurs in a chemical reaction. So our introduction for this is we're going to be learning about some special chemical reactions today. Scientists can tell when a chemical reaction has occurred because one of the following will result. There's going to be fizzing. There could be a color change. There could be a temperature change. There's a creation of a totally new product. Okay, and now this product that is made cannot be changed back into the original ingredients. So keep that in mind. All right. We're going to jump right into our activities today with activity number one. Catalase decomposes hydrogen peroxide. Okay, activity number one. Catalase decomposes hydrogen peroxide. So, first things first, we're going to talk about four words. We're going to talk about the word catalyst. We're going to talk about enzymes, catalase, and decomposition. Okay? A catalyst is an enzyme that speeds up a chemical reaction. Okay, so it makes it go, it makes it happen a little faster. An enzyme, enzymes are proteins that are in plants and animals. And what they do is they help biochemical reactions to occur. Enzymes build up and break down other molecules. Catalase, it sounds like catalyst, but it's close. Catalase is an enzyme that is contained in all living organisms, and it helps break down hydrogen peroxide, or it helps the decomposition, if you will. Decomposition is the breaking down or the separating of things through a chemical reaction. So what we're going to do is we're going to take a look at how that works here. So what we have here on our plate is we have a tasty banana slice, and then we have this liquid here. Science Explorers, let's take a second to observe this liquid. We're not going to taste it because that's dangerous in science. We don't do that. But we can use our eyes to look at it. It's clear. I mean, it very well could be water. But again, even if you think it's water, don't go drinking it because it could be something else. You can do the waft test where you can smell it. Doesn't smell like anything like vinegar or anything. It's not giving off any odors like you would get from alcohol or vinegar. What's actually in this cup is hydrogen peroxide. Now, hydrogen peroxide, if you want to come down here and take a look, hydrogen peroxide is actually the 3% solution of hydrogen peroxide, which means that it's 3 parts hydrogen diluted with 97 parts water. So that's what we have here in our cup. Again, we have a banana. We're going to take our banana and we're going to use our plastic fork to just kind of, mash up our banana a little bit, okay? We're going to get some mashed banana. My friend Helium Hayden loves to mash up her bananas. When she eats it, she calls them mashed bananas, kind of like mashed potatoes, if you will. So we mashed it up a little. There. Okay. So, there we have that. Now, what we're going to do is we're going to take some of our hydrogen peroxide with our pipette, and we're going to drop some on the banana. We're going to put some drips on the banana. What I want you to do right now is pause the video and make a hypothesis of what is going to happen when I put some of our hydrogen peroxide on our banana slice.
go ahead and pause the video. Okay, welcome back. Here we go. We have our pipette. We're going to take some of our hydrogen peroxide inside, and we're going to drip some on. Let's see if our predictions were correct. Okay. Huh. Let's see. Yeah, maybe Data Dietrich didn't smash it up enough. It is fizzing a little bit when you see it there. When I'm kind of mixing it together now, you can see that it fizzed a little bit. Okay. The hydrogen peroxide will fizz when it comes out and into contact with it. And you do kind of see it happening right now. If you get close, it's getting white and frothy. That's the fizzing that occurs. It's not like a carbonated soda or anything, but it is definitely doing some fizzing action there. Okay. Fizzing is one sign that a chemical reaction is happening. This is because the cells of the banana contain catalase. If you put hydrogen peroxide on a fresh cut, for example, if you cut your finger and you put hydrogen peroxide on it, it will fizz because our cells also have catalase in it. Catalase is the catalyst that breaks down hydrogen peroxide. We know that catalase is present in the banana because of all of this fizzing. Check that out. Like, that is definitely different. Interesting stuff, right? If you take a look at our hydrogen peroxide bottle here, okay, it's interesting that hydrogen peroxide comes in this nice brown bottle compared to, let's say, rubbing alcohol, which comes in a clear bottle, right? It's different. The reason why the hydrogen peroxide bottle is dark like that is because hydrogen peroxide breaks down naturally over time, and it breaks down into water and oxygen. Air, light, and heat contribute to its decomposition, and that's why hydrogen peroxide comes in this dark bottle. Isn't that interesting too? Hydrogen peroxide is used by humans to clean wounds and also to clean teeth and gums sometimes, which was kind of shocking to me. It is also used to clean water in sewage plants. The cells in the body make hydrogen peroxide while converting sugars into ATP, or cellular respiration, the main source of energy for all living organisms. However, too much peroxide would become toxic. Our cells cope with that using an enzyme called catalase, right? And it helps break that down into the water and oxygen into our bodies. Okay, so if you remember that the cells of every living thing contain catalase, so that's important, right? Let's move on to our second activity where we're going to be reusing some of our stuff over here, like our hydrogen peroxide.
Activity number 2, catalase decomposes hydrogen peroxide part 2.
Okay, here's part 2 of catalase decomposes hydrogen peroxide. The first thing we're looking at, we still have our hydrogen peroxide cup, right? And now we have another solution, okay? It's kind of murky, kind of cloudy, almost a beige or tan kind of color. What's inside here is actually a yeast solution that we mixed up, that we also let bloom a little bit for about 10 minutes before we started this activity. So what we're doing, well, let's talk about yeast first, okay? Yeast is actually a living organism. So this is what yeast looks like, and it's actually a dried, and it's not active right now. The way to activate yeast is to use a liquid. In this instance, it was warm water. Thank goodness for Data Dietrich's wife. I made the water too warm, and I killed yeast at first. But yeast is a living organism. it's what we use in breads to make our dough rise, because what happens is the yeast, being a living organism, will eat the sugars in the different mixtures, and what happens is it poops carbon dioxide, and that's what the bubbles are, and that's what makes the bread rise, and then it gives you those little airy pockets inside of bread, right? Crazy. So the single-celled yeast actually digests the sugar and the dough. It gives off the carbon dioxide in the process. The bubbles are CO2 in the bread. That's what gives it its texture. Since yeast is a living, it should contain, that's right, catalase, right? So how do we figure that out? What we're going to do is we're going to take our yeast and we're going to add it to the hydrogen peroxide. What do you think will happen when I add the yeast mixture, a living organism, into our hydrogen peroxide based on what we know from the first activity? Take a pause and talk to a neighbor about this. All right, here we go. So, what do you think will happen? Okay, I'm thinking, since it's living, and I know that it contains catalase, it should fizz. So, I don't think I need the pipette here. I think I'm just supposed to take it, and I'm supposed to pour it inside. Here we go. 3, 2, 1. Okay, let's see what's happening here. Oh, can we get that real close? Do you see the fizzing happening? Okay, there it is. It's starting to fizz. That's proving to us scientists that there's a chemical reaction happening with that hydrogen peroxide and that yeast mixture. Look at that. Look at that. That's amazing. There's all that fizz. It's like a nice little foam on top. This reaction is going to last for about a minute. Okay. When the fizzing is over, you could probably, which mine is still fizzing. Whoo-wee. It's about a minute. It'll take about a minute, but when it's over, you can use your pipette or a craft stick to stir it around. And what that's going to do is it's going to help the remaining peroxide decompose. What's happening right now is the yeast, the living organism, and the catalase is helping decompose the hydrogen peroxide. It smells like a bakery in here right now. It's pretty it's pretty interesting. And when you do finally get it to stir a little bit, okay, what's going to happen is the rest of that yeast and peroxide will interact, and it might fizz some more. The hydrogen peroxide right now, what's happening is it's being broken down into water and oxygen. Okay? When this is all done, we'll move on to activity number 3, and we're going to see if the hydrogen peroxide is still there. Oh, this is interesting, right? Yeah, it's a fun way to stir. Ooh, I ruined my fizz. Anyhow, we're going to move on. And activity number 3 is all the hydrogen peroxide decomposed.
Activity number 3 is all the hydrogen peroxide decomposed.
All right, here we go. Activity number 3 is all the hydrogen peroxide decomposed. So we have our mixture of our hydrogen peroxide, which was hydrogen peroxide, and we poured our yeast mixture in there. We have a new slice of banana. We have another fork again. So again, we're gonna sit here and we're gonna mash up our banana. I'm gonna do a better job at mashing it up this time, I promise. I guess I didn't mash it up enough last time, so we're mashing up. Oh, neat. Don't make a mess, I guess. Probably a bad idea. Mashed banana. All right, so if you remember, I said what happened was the yeast, the living organism, helped speed up the decomposition of the hydrogen peroxide. It helped break it down into that water and oxygen. The oxygen was being released as those bubbles, and the water just was in our mixture there. That's what was left over. So what we're going to do now is we're going to see, is there any hydrogen peroxide that's actively left in there? Because when we took the hydrogen peroxide before and we put it on the banana, it foamed, right? We're going to take a pipette full of this liquid, okay? And we're going to put it on our banana. Ready? 3, 2, 1. Okay. What do you think? Oh, man. All right, so hold on. Let me see. You stay there. I'll get the old one out. This is the old banana. It fizzed, and it looked all... And now that's what it looks like. This is the new banana. This was the hydrogen peroxide. This was the yeast hydrogen peroxide. What do we got going on? There's not foaming going on, right? It did not fizz. The peroxide has been decomposed, okay? This shows that the catalase in the yeast helped to break down that peroxide into that water and oxygen. So the catalase in the yeast sped up the decomposition of the hydrogen peroxide into water and oxygen, substances that are safe for living organisms. The fact that there was no fizzing on this second banana, okay, that shows that there's no hydrogen peroxide left. So the chemical reaction completed the decomposition or the breaking down of the hydrogen peroxide into what? Again, water and oxygen. Let's move on to our next activity, activity number 4.
Activity number 4, catalyst remains unchanged.
All right, here we go. Activity number 4, catalyst remains unchanged. All right, so a catalyst, well, catalysts are substances that speed up chemical reactions. A true catalyst does this without being part of the reaction, right? So that means that it's not all used up. A catalyst can be used over and over again since it's not used up in the reaction. Okay, recall the catalyst inside the living organism. It's called an enzyme, right? Catalase is an enzyme that is used over and over again in our bodies to break down hydrogen peroxide that our cells produce every second of every day. Without hydrogen peroxide produced during cell respiration would poison us. Right? And our bodies are truly amazing that it can regulate that kind of stuff. The next experiment will show that a catalyst in our last reaction remains unchanged. Now, our catalyst was what? Was that the yeast? Right? The yeast is still active even though it broke down the hydrogen peroxide. In order to demonstrate this is what we're going to do. We're going to make a simple dough. Okay, so we still have our hydrogen peroxide and yeast combination here. The hydrogen peroxide is gone into water and oxygen. What's left is that yeast mixture, and the yeast, what we're trying to prove, is still active. So we have some sugar, and what does yeast eat? Well, yeast eats sugar. We talked about that earlier. So we're going to take this packet of sugar. We're going to put it in here. Okay. Now we gave the yeast some it's, snack. Mix it up a little bit. All right. Daddy Dietrich's getting kind of nervous because I have to add some flour to this because now we need our flour to make a dough, right? So we're going to take our soup spoons here and we're going to add 1 leveled soup spoon. We're going to try to level this one off again and say 2. All right, let me get a little stir in here. It's a good thing I have this plate down, right? Oh boy. I think I had too much fluid, to be honest with you. Let's go 3. Oh boy. Data Dietrich's running out of room here. And it's still too liquidy. All right. I've got to get it to the consistency of what looks like cake batter, but this is definitely still too runny. All right. Supposed to only have to do 3. Let's see what happens if I do 4. Maybe I'll get a bigger cup. Thank you to my camera person. All right, here we go. Whew. And I'm just gonna add another scoop of flour. This is science, kiddos. This is why we're here. Things aren't always gonna work out right the first time, but we'll figure it out. I think I'm up to 4 spoons now. It's getting thicker. It's starting to become more like that cake batter that we're looking for. This isn't something that we're going to be eating anyway, right? Considering the fact that we started with a base of hydrogen peroxide. Although it did get broken down, we still don't want to eat something like that. I think 1 more will do it. 1 more. Yeah, there we go. Now it looks like I can pour some pancakes with this. What do you think? What do you think, Science Explorers? Made a little bit of a mess on my tray. Whoopsies. All right. Now, next thing. I'm going to leave it at this. We're going to see if it works. Next thing is you're going to take a permanent marker. And you're going to mark on the side right here. For this activity to prove our point, what should happen is the yeast, if it's still active, if it's still alive, if it's still a living organism, and it's still a catalyst, it should make this rise, okay? We should see our yeast will eat the sugars and excrete carbon dioxide and make bubbles and make this rise, okay? We're gonna check back in on this activity later after we move on to a couple other activities. Let's check out what activity 5 has in store for us.
Activity number 5, chemical formulas and equations. All right, here we are with activity number 5, chemical formulas and equations. This is where we're gonna have some fun making some models, okay? So, what are we talking about when we talk about chemical formulas? A chemical formula is a model used to show like the recipe of a chemical substance, right? For example, H2O. H2O, if you've ever heard of that, it's water, right? But it's telling you the chemical makeup of that molecule. It's 2 parts hydrogen, 1 part oxygen. Okay, a chemical equation is a model used to show the molecular process of a chemical reaction. And then we have atoms and molecules. Atoms are our building blocks, right? They're the smallest units or the smallest elements of our physical world. And then they form together and they group together and they make up molecules. Okay, so what we're looking at to do here in this activity is we're going to make some models of our chemicals here. We have our hydrogen peroxide, right? And we're going to try to model what happens when it decomposes or breaks down into our water and our oxygen. Right? So we're going from 2H2O2 to 2H2O and
O2. And what happens is we're going to model out the fact that we start with all these pieces over here, and then we're going to end up with the same amount of pieces on this side, just broken apart then, right? Or decomposed. So we're going to be working with some model magic here. Science Explorer instructors, the magic the model magic that you got more of the color of, that's what you're going to be modeling the oxygen with, and the hydrogen will be modeled with the one that you have less of the color with. What I'm going to do is I'm going to model the oxygen with my black metal magic, model magic. There it is. Oh, it's not black, it's white. Silly Dietrich. All right, you're gonna get about half of it. Okay, actually, you know what? I think I changed my mind. I'm gonna use this for the hydrogen. I'm gonna use the red for the oxygen. All right, so what happens here is you get your model magic, and you're gonna have to make 8 pieces out of this, okay? So it's probably best to break it in half, and break each one of those halves in half, right? And then, to get 8, you have to break each one of those in half again, okay? So there's 4, there's 6, and here's 8. We're gonna take them, we're gonna roll them into our little atoms of hydrogen, right? I'm gonna have 8 pieces of them, okay? 8 pieces. There's 4. You got to work kind of quickly, because if you don't, they dry up and you won't be able to mold them anymore. It's magic. It's science. So now that I have, anybody ever rolled a meatball before? I have. Made meatballs just today. Don't eat these though. Okay, so I have my hydrogen. Now I'm gonna take my red and I'm gonna make my oxygen. You need 8 pieces of the oxygen as well. You're gonna get from your Science Explorer instructor about half of this pack of this color. And you're going to do the same thing, where we're going to break it in half. Okay, we're going to break those in half. We're going to break those in half. Okay. I feel like I was a little cheap on that one. All right, and same thing. Rolling our atoms of oxygen out. All right. Check it out. We're a rolling factory now. That one seems a little tiny too. And since I can, I will take a little extra there. All right. And there we go. Now I have all of my oxygen atoms as well. So to model our 2H2O2, or hydrogen peroxide, we're going to take 2 oxygens and 2 oxygens, right? There's O2, 2 oxygens. And we're going to take 2 hydrogens and 2 hydrogens. So we have 2 H2, right? O2. 2 pieces of them, 2 molecules of that. There you go. Right? Now, when that gets broken down, or when it decomposes, we get 2 H2O. Okay? So there's one single O for each of the two, right? And each one of those gets little ears. No, little hydrogen pieces. So each one of them gets 2 hydrogen, 1 oxygen. And there's 2 parts of it. The last thing you do is you have O2. This is the oxygen that has escaped from the decomposition of the hydrogen peroxide. You just take your 2 oxygen atoms and you put them together like that. So we went from 2H2O2 to decomposing into 2H2O and plus O2, which is oxygen. Now, what would happen here now is, now that you have your model all set, you're going to take some glue, right, which Dietrich should have poured earlier. Yeah. We don't need much. Okay, Science Explorer instructors, you'll hand out some glue and some Q-tips. And to make our model stick together, we'll take a little dab of glue. I guess we don't even really need to do it, but probably should have did it before. If they're stuck together nicely, I guess you don't even need to mess with the glue. Yeah, I think I stuck them together pretty good. So we're just going to stick them off to the side. Last part of our activity here for activity number 5 with our models here is, let's count up our number of atoms. So how many hydrogen do we have on this side? We have 1, 2, 3, 4 hydrogen total, and 1, 2, 3, 4 oxygen total. After it decomposes or it breaks down, how many hydrogen do we have over here? Well, we still have 1, 2, 3, 4 hydrogen. And guess what? We still have 1, 2, 3, 4 oxygen. They're just broken apart and decomposed into different things. All right? Pretty amazing. Okay? So that is it for activity number 5. We're moving on to activity number 6. Activity number 6 sounds fun. Called foaming fountain.
And activity number 6, foaming fountain. Okay, capturing the oxygen in the decomposition of hydrogen peroxide.
All right, Science Explorers, here we go. We're doing our foaming fountains. This is where we're going to try to capture the oxygen that's being decomposed when we pour our yeast solution into our hydrogen peroxide. Remember, it fizzed a lot. What we have going on is we're going to use dish detergent, right? Or soap to help try to capture those oxygen bubbles. We're going to use some food coloring just to give it some color. We have our yeast solution that we made earlier, and we have a bottle of our hydrogen peroxide, which will not be hydrogen peroxide. Science Explorer instructors, you guys can set up a station, or you can walk around and add this stuff to their mix for them. If you have older Science Explorers, you might want to be able to do that. They need about a tablespoon of the dish detergent, so I'm just gonna give a nice healthy pour here of that. And a little more. That's good. Yeah, what do you think? Okay, a couple drops of food coloring. I picked green. Might make yellow, right? Ooh. Ooh. Or that wouldn't make yellow, but so there we go. We have that all set up. Okay. Now what we have to do is we have to make this kind of like a give it a little bit of a bend to give us a little pour spout here, because it's going to be hard to pour it in otherwise. So just kind of get a little point there. And we're going to see what happens, right? We're going to pour the yeast mixture into our hydrogen peroxide, soap, and food coloring. Ready? Here we go. Wow, that was a pretty good pour. boy. Oh, here it comes. Whoa. What is happening right now? Uh-oh. Hopefully that tray is deep enough. If not, Dada Dietrich is going to grab some napkins. What's happening with this? There's the soap is actually capturing the oxygen and causing to catch some of those bubbles, right? The breakdown of the hydrogen peroxide creates the little fountain of foam, as you see. Smells nice and clean. Doesn't smell as yeasty, but it's just going to keep going. Oh, wow. You hear it fizzing a little bit, okay? And you definitely see the foam kind of oozing out. This is really neat, yeah? It's not harmful, and if you touch it, it actually will be a little warmer than you think. It's not so warm that it's hot, but what's happening is there's a chemical reaction happening, and this chemical reaction is actually exothermic, meaning that it gives off some heat. It's not a lot of heat, but it gives off some heat. Interesting, right? It's still going. It's still going, and it's kind of rising. Wow. All right, so there's our foaming fountains. That was activity number 6. We're going to check in on our yeast creation, our dough, before we head out for the day. Okay, guys, here we are to wrap up our lesson today of super quick catalysts. We're back here to check on our dough. So if you look down here at our dough, yep, sure, the dough did go above the line, okay? What happened was that even after the yeast decomposed the hydrogen peroxide, it was still there. The catalyst of the yeast was still there. So it was still able to function, okay? It did not deplete after it decomposed the hydrogen peroxide, which is what our dough lesson has proven. You guys will get lids for these cups. You guys can close them up and take them home with you, as well as your fountain will seal up. You can close that up, and your molecules. So I hope you guys enjoyed the lesson today. I hope you learned a lot. And remember, here at Science Explorers, if it's not fun, we're not doing it.